Hello, this is Ed Chapman, and this video cast is the third in a series on respiration. And in this video cast, we're going to focus on the process called glycolysis and another process called the citric acid cycle. Here's a big picture of how these fit together. We're going to start with a food molecule like glucose, which of course is a 6-carbon molecule. And we're going to run it through a process called glycolysis, which is the first step. And out of that, we're going to get some ATP energy and some NADH. Remember, NADH is an electron carrier molecule that is carrying 2 electrons and holding onto a hydrogen ion in the process. The glucose is going to be converted into something called pyruvate. And pyruvate is a 3-carbon molecule. So if we're going from 6 to 3, there are actually 2 of these guys. So we're going to get 2 pyruvates. And then the pyruvates are going to feed into something called the citric acid cycle and be converted to carbon dioxide. And of course, if we're breaking something down into carbon dioxide and we start with 6 carbons, we're going to have to end up with 3 carbons. So one of the things you can do here is just follow the carbon counts. We start out with 6, break it down to 3, and break it down to 6 molecules, each containing 1 carbon apiece. So carbon dioxide, of course, is 1 carbon per carbon dioxide, so there's 6 of these guys, 6 CO2s. All right, and the citric acid cycle is also going to produce some ATP, some NADH, and a new electron carrier molecule called FADH2, which, if you want to read about it in the book, it's a little bit different from NADH, but we're going to treat it basically the same here. Okay, glycolysis, which is the first step, takes glucose and breaks it down, and the word tells you what it means. Glyco refers to sugar, sounds a lot like glucose, and lysis, like you may have heard in lysosome, means to break. So glycolysis literally means to break sugar. And it's performed by all cells, as far as I know, prokaryote and eukaryote, which makes it a metabolic pathway that's very old. It's probably been around a long time, at least 4.6 billion years since bacteria first appeared. And it's a metabolic pathway made up of many different enzymes working together, each one using the products of the ones before it as reactants. What glycolysis does is it splits each glucose into 2 pyruvate molecules, and it's something that takes place in the cytoplasm. You don't need organelles like mitochondria to do that, hence it being something that prokaryotes do. Because remember, prokaryotes don't have mitochondria. All right, glycolysis is an energy yielding reaction, so it's a net exergonic reaction, which means it's going to be downhill. But the first step in glycolysis is actually uphill, because we've got to take glucose and raise its energy level up to a point where we can call it an energized state. And then this energized state of glucose can then fall downhill, so to speak, to the pyruvates I mentioned earlier. Now, if you look in your textbook, there are lots and lots of steps happening here and lots of steps happening here. You're not responsible for knowing all those different steps. And this molecule here, the energized state, it has a name, but again, I'm not worried about you knowing the name for that. Now, the raising up of glucose to the energized state is, of course, uphill. So it's going to require some energy, and that energy is coming from 2 ATPs. So we have to invest 2 ATP molecules to get it to this point. All right. Then the bigger downhill fall is going to produce 4 ATPs. All right. So changing this energized glucose to the 2 pyruvates is going to produce 4 ATPs. So per glucose, we actually have a net gain of plus 2 ATPs. All right. If we do the accounting here, we've got 2 ATP cost and a 4 ATP benefit. It comes out to a plus 2 ATP gain. And we also get, from the downhill slide, 2 NADH molecules being produced. Now, here's the numbers. For glucose going to pyruvate, the payoff for this reaction is a little bit of ATP and a little bit of NADH. Okay? Now, the pyruvates are then going to be fed into something called the citric acid cycle. Now, pyruvate has 2 possible fates. We can metabolize it using oxygen and completely oxidize it down into carbon dioxide. That's going to give us the most energy. Or pyruvate can be fermented. It can be run through a different set of enzymes that don't require oxygen to do their job, and we can get some energy out of that also. And there are two types of fermentation. One type produces ethanol, which is the food alcohol you probably have heard about, or lactic acid, which is an acid found in things like yogurt. So we're going to cover the aerobic side, or the citric acid side, first, but we're going to save that for the next video cast. Thank you. Thanks for listening.